Cellular respiration is a process in which the cells convert glucose and oxygen into energy, carbon dioxide, and water. It occurs on the cytoplasms, and it starts with the process of glycolysis, which is the process in which glucose, a 6-carbon molecule, breaks down into 2 molecules of pyruvate, which is 3 carbons each. This process requires 2 ATP molecules to start, and then those 2 ATP molecules become 4 ATP molecules, and besides that, there is NAD that becomes NADH. So the net gain of glycolysis is 2 ATP and then 2 NADH and the 2 pyruvate molecules. After glycolysis, then each pyruvate molecule is transported from the cytoplasms into the mitochondria, and then once in the mitochondria, the pyruvate goes into the carbonylation, which means that one atom on carbon is removed from the pyruvate molecule as carbon dioxide, and then it becomes a 2-carbon compound. But during this process, electrons are also transferred to NAD plus to NADH, and then this NADH will be used later on the electron transport chain. Then a coenzyme goes into the process, and the two remaining carbons combine to this coenzyme, and they create a molecule called acetyl-CoA. This 2-carbon molecule then will be used into our next step of cellular respiration, which is the Krebs cycle. In the Krebs cycle, acetyl-CoA combines with a 4-carbon molecule called oxalacetate to form citrate. And in this step, the coenzyme gets removed and it becomes the citrate. Then citrate is converted into its isomer isocitrate to a series of rearrangement into the molecule structure. So there's still 6 carbons in there, but then it's just rearranged. Okay, so then isocitrate is oxidized and it loses 1 carbon molecule in the way of carbon dioxide, and during that process, NAD plus is converted into NADH. This creates a 5-carbon molecule with a funny name. Then this 5-carbon molecule gets oxidized, and then it becomes a 4-carbon molecule. And during this oxidation, it also loses 1 carbon in the way of carbon dioxide, and then NAD plus is reduced to NADH. On the next step, then GTP is produced through substrate level phosphorylation, and this reaction is catalyzed by succinyl-CoA synthesis. And then the succinate is oxidized to fumarate, and during this process, FAD gets converted into FADH2. Okay, then fumarate is hydrated to form malachite by the enzyme fumarase, and then the final oxidation happened when malate is oxidized back to oxacellate with another production of NADH, and this step is the final step of the Krebs cycle, and because the oxalate is the same molecule at the beginning of the Krebs cycle, and so it just repeats. In summary, the products of the Krebs cycle will be 3 NADH, 1 FADH2, 1 GTP, and 2 carbon dioxide. Then we move on into the inner mitochondrial membrane of the cell, in which the electron transport change occurs. And this begins with the electron donors, which are the NADH and FADH produced on the Krebs cycle and glycolysis. Okay, so as electron passes through the complex proteins, they release energy, and then the energy released from this electron transfer is used to pump protons or hydrogen ions from the mitochondrial matrix into the inner membrane space. This creates a proton gradient across the inner mitochondrial membrane. And then we move on into the process of chemosmosis.